I'm sorry. Our next speaker is Christy Landis from Rice University, and the title is Super Resolution Spectroscopy Reveals Molecular Scale Detail in Ion Exchange Protein Separations. Okay, Christy. Thank you.
Thank you, Mary Ellen. First, I would just want to make sure that I say what an honor it is to be included in this symposium with so many great speakers. And thank you also, Mary Ellen, for including me. And Mary, I'm very honored to be here. As a spectroscopist, starting to transition into understanding systems that are being discussed here. So that means, first of all, we should say congratulations and very much well-deserved to Mary for this award. And as well as just being in the room with you today to give the talk, I think it's nifty to be able to acknowledge that you've had, probably you don't know, but a strong impact on my career already, albeit short. My very first talk that I gave at a national ACS meeting as a faculty member, I was talking about a paper in which we had referenced this early 2001 paper that you'd written in applied spectroscopy, and about transport in soft interfacial materials, which is how kind of I got here. And right before my talk started, my student who was in the audience ran up to me and whispered in my ear, "Oh my God, Mary Worth's here," in the audience, which didn't help calm my nerves at all. But you were so gracious and nice to stay after and talk to me and give some insight about the single molecule experiments that we were doing to try to understand transport at these nanoscale interfaces. And that was even before we started thinking about transitioning into applying some of our methods to understand processes in chromatography. So that was very, very early, and I'm happy to have an even stronger, I think, relationship with you as well. And I would also like to thank the students who participated in the work that I'm going to tell you about today. In particular, Lydia Kisley is now a fourth-year student and has aspirations to a faculty position, and she certainly has the capacity to do it. So I will be advertising to send her to a postdoc at some point soon. So if you want someone way smarter than me and way more organized than me and way harder working than me, even in your group, Lydia will be looking for a postdoc soon. And I should also mention that this project is a funded collaboration between myself and Richard Wilson, who's a bioengineer in separations at the University of Houston. And it couldn't be possible without that collaborative effort, because I have definitely started my career as a physical chemist and spectroscopist, and so we spend our time, whether we know it or not, worshiping at the feet of the Gaussian energy structure profile. And that's true even if you're a synthetic chemist. You want one thing and only one thing in your filter frit, or if you're a spectroscopist like me, you're studying one transition, or even if you're a theoretician, you know when your simulation's finished when it's minimized. So all of us live in this realm, but I've been very interested in my own career in systems that deviate not just a little bit from a well-defined structure-function relationship, but instead have a completely different model for how their structure and their function are related. Lots of natural systems, instead of having just one overall structure, have multiple structures. They have multiple intermediates that might combine to exact a function. That's called redundancy, and even completely counterintuitive to how we would design things. Often degeneracy is also built in, so there are different pathways to different products, but that's how nature does things like allow us to have a gene that codes for a particular disease but not get the disease. There are multiple pathways and multiple intermediates all combining and competing for overall function. And from an experimental physical chemistry perspective, there are all sorts of interesting challenges to understand these systems, but also I think that there's, as has already been discussed today in the previous two talks, understanding the role of heterogeneity in an overall macroscale function has applications to synthetic materials technology as well, and that's what I'm going to tell you about today. But that means in studying these types of systems, we spend a bunch of time doing biological benchmarks to find this type of behavior. We spend a lot of time developing methods to even understand what the heck we have in terms of the data. And then, as I mentioned, we would like to look for this type of function in synthetic material systems. But the story that I'm going to tell you today kind of bridges all three of these pieces here. But just to make sure that we all agree, one of the reasons that we so like working with these types of systems is that you don't have to collect all the data to completely reconstruct this landscape. You need to just collect enough to fit a Gaussian. There's only 2 parameters for a 1D Gaussian and 3 parameters for a 2D Gaussian. And so it's easy experimentally to fit. And that's what happens all the time with chromatographic separations, that you get your elution curve and you fit it to some version of the Van Diepter equation or something else. But in these types of systems, of course, you can't fit to any particular equation. There isn't a theory usually. Okay, so you don't have a theory to fit to, but then you would have to collect Avogadro's number of data points to be able to collect this all experimentally, and that's not possible either. And then there's all sorts of interesting, just practical consideration. How do you tell the difference between local fluctuations and just noise? And we spent a lot of time in the first, I guess, 7 years of my independent career trying to figure out how to address those systems. And so in case anybody's interested, we have a bunch of papers on really just math, applied mathematics, and how to use it to apply to these types of systems. But that's not so interesting and related to what I'm going to tell you about today. But we make a bunch of MATLAB code available for anybody who cares about that type of thing. So for example, underneath some of the things that I'm going to have to tell you about today is the need to do particle tracking, but in noisy and messy and heterogeneous systems where you have intermittency or variances and intensities and things like that, and to understand transport in charged and crowded environments, such as you might have at the interface between a stationary phase and a mobile phase. So we spent some time worrying about that. And then additionally, you'd like to be able to resolve interactions between your analyte and the adsorbent at a resolution approaching the size of the analyte. And so we've done some development of super resolution methods to image biomedical and biomaterials applications. And that's the piece that relates most closely to what I'm going to tell you about today. I don't have to motivate how important protein separations are to this audience, but in case no one has ever used this particular graph to talk to an audience, I thought I'd still keep it up today. At the University of Arizona, this Jardardson makes a giant poster-sized PDF every year of the top 200 drugs. So this is a slide and an eye test, okay, simultaneously. This is the top 200 drugs sold globally in 2006. And you can't recognize anything here, but hopefully you can see that everything up here, except for a couple things, are in black and white. And that's because everything up here in 2006 was what we would consider an organic or some sort of combinatorial chemistry type material. And there's even, there's a couple brightly colored things here, but that's just salt. That's also a chemical, right? But when you move from 2006 to 2012, the stark difference between the types of materials that are being used for pharmaceuticals is very obvious, I think, when you use this big picture graph here. And so these are available every year at the University of Arizona website if anybody would like to use it. And so you can see here that very quickly and very abruptly the industry has transitioned to use biological materials such as proteins and DNAs, and all those materials present their own unique and interesting challenges in terms of the separation, and some of those Mary already talked about earlier. And what we would like to do is we would like to understand chromatography at the molecular scale so that we could describe it from a mechanistic perspective. So to relate in some way the empirical nature of the Van Diepter equation and all of its parameters, as Mary mentioned before, to something mechanistic so that we could move away from ever having discussions like your experimental plate height makes no sense. And the reason it makes no sense is that this equation is completely empirical, and if all of the mechanistic contributors there could be described, then it would be easier and more appropriate to tune all these components here and understand them and not be so shocked when we discover a completely new mechanism that's contributing. And some of our inspiration came from this paper from Wu and Walters that was published in 92, I believe, where, and using some more standard techniques, of course, because it was 92, in terms of ion exchange, which should be purely a coulombic and electrostatic interaction between the analyte and the column, that there's some sort of denticity there, a multivalency more appropriate to something like affinity chromatography, So there's something more complicated going on there with multiple binding type of interactions between a protein and the ion exchange column. But we like to be able to look at this at the level of understanding how an individual protein and individual ligand interact. And coming back to then that graph that I showed on my first slide, what I mean is that at our most basic interpretation of how chromatography works is that there's assumed to be a weak interaction that is multiplied over the course of the column, and that weak interaction then could be described as a single Gaussian, and that leads to a perfect elution curve. But of course, as Mary said, that's never the case, especially with proteins. And if we're thinking mechanistically, we might imagine that underneath the fronting and tailing that happens in these peaks, there's actually multiple processes that are going on, including these very rare strong events that Mary talked about at the surface of the silica. And if it were possible to understand at the mechanistic level each one of those types of interactions, you could tune in the mechanism that you would desire and tune out all the others by doing things like polymerizing at the surface of the silica. And of course, you like to do that in terms of the more complicated and heterogeneous protein structure. So that's our goal, to understand the underlying heterogeneity that leads to the broadening in elution peaks. And of course, as spectroscopists, we'd like to use our tool set or even develop a new tool set that would be appropriate for understanding that. And so based on that Wu and Walters paper and also some preliminary work by in the Wilson group at UH, we're going to hypothesize that one of the methods for preparing ion exchange ligand packing is going to lead to a hidden phenomenon, and that is, as was confirmed in my visit to 3M 3 weeks ago, that how you optimize your column is that you choose your ligand and you run an elution, and then you increase the packing density, and you run an elution, and then you increase the packing density, and you keep going iteratively until you've empirically optimized, and then eventually that tails off again. And so based on how things are done, and also on that Wu and Walters paper, we're going to hypothesize that what's actually going on at the mechanistic level within these ion exchange columns for proteins is that the packing density is so high that the ligands can overcome the electrostatic repulsion between them and cluster together. And it's the clustering, random clustering, that's being optimized by tuning the loading density. And then eventually, of course, once you load up so high, you actually create aggregates, and then that breaks down the A parameter in the Van Diemter equation. So in order to test that hypothesis, though, we're going to have to do the opposite of what you would do in chromatography. Instead of increasing the loading density to improve the separation, we actually want to decrease the loading density so that we can be absolutely sure that we can understand and measure the interaction of a single protein with a single ligand. And then, ideally, we'd like to be able to use our newest spectroscopy method, super resolution microscopy, to resolve individual proteins binding with individual ligands to make sure that we only have single ligands. And then, because we have the ability to resolve these individual adsorption, desorption interactions, then to reach a mechanistic understanding, instead of collecting things like equilibrium binding coefficients, ideally you could collect the association and dissociation or binding and unbinding rate constants separately and then put those together to make an equilibrium constant. And then finally, we could choose either to relate this to the Van Diemter equation, but given its purely or maybe corrected empirical equation, we'd also like to relate our mechanistic data to a pure mechanistic theory, a stochastic theory of chromatography that really relies on true molecular scale interactions. And luckily, we don't have to derive that equation. People that have already been discussed earlier today have derived that equation. So in 1955, Giddings, who was already mentioned, and Eyring at the University of Utah published a beautiful stochastic theory based on statistical mechanics that was just, kind of taking off at that time, of the theory of ion exchange chromatography. And they published several improvements on it based on individual rate constants for the interaction of a single protein with a single ligand site, and the time that that particular protein would spend in the mobile phase, and then a continuum of those types of interactions over an entire column. It's a very elegant and beautiful theory, but unfortunately it relies, in order for it to be applied, you have to actually extract these rate constants, and so you can't use equilibrium data. You need the dynamic information. So even though this is a very beautiful paper published in 1955 by people that are very highly respected in the field, it has fewer than 100 citations even to this day, because it's not easy to get the data out to put in this equation. But fortunately, we don't have to derive it. Smarter people than us already have done so. We just need to find a way to extract this information and get the rate constants. And so how are we going to do this? I already said the first thing we're going to do is reduce the loading density of our ligand in our stationary phase so that we can resolve single interactions. The other thing we need to do is we need to move from a three-dimensional column geometry for our stationary phase and mobile phase interaction to a 2D thin film. So we're going to have a thin film of our stationary phase support, which in this case is agarose because it's easy to spin cast and it's easy to use chemistry to control the pore size of the agarose. And then that's going to be decorated with common ion exchange ligands, either single isolated ligands, as are used commercially, or to test our clustering hypothesis, we're going to actually engineer clusters of our peptides by making chemically a polypeptide so we can control the cluster size and position. We don't allow it to happen accidentally. And then our model analyte is going to be a globular protein alpha-lactalbumin so that we can avoid any funny business with respect to the protein size and shape in terms of its transport. But it does, like all proteins, have it has a heterogeneous distribution of charged peptides on the surface and in the interior of this protein. And in particular, it has this 5-amino acid calcium binding pocket. So we might expect, based on the disparities in the charge distribution, if there is some sort of denticity in how a protein could interact with the analyte, that there might be differences there. And for our adsorbent phase, we're going to use one of the standard positively charged ligands, which is the guanidinium group on an argininimide. And so we're going to either use monoargininimide, the commonly used ligand, or we're going to use a penta-argininimide that's been pre-polymerized so that we can control the spacing of the charges. And how we're going to do the experiment is on what ordinarily would be a traditional wide-field microscope in which you use the objective to do total internal reflection. And the reason that we do that is that when you use total internal reflection wide-field excitation, the evanescent field that's caused by the laser only goes about 100 nanometers into the sample. And so that means if you're interested in the interface between the mobile phase and the stationary phase, you only see that interface and nothing else. This is really low background measurement. And so then our thin film sample goes on top here, and then we flow over our mobile phase, and with our microscope, with this total internal reflection geometry, we only see about the 100 nanometers right where the stationary phase and the mobile phase come together. And then how do we resolve anything on the order of the size of a protein? Starting about in 2006, maybe 5 different groups on the planet came up with different ways to do super resolution microscopy, which is to image below the diffraction limit of light. There's not really time to tell you about the other methods, which include lots of different acronyms like STED and PALM and STARM and RACM and SMACM. But the one that I am interested in applying here is most related to the one that was developed by Robin Hochstrasser, who unfortunately he was a brilliant physical chemist and spectroscopist, but unfortunately our community lost him last year. He died kind of unexpectedly. And his method for doing super resolution that's called PAINT, point accumulation for imaging and nanoscale topography, it is very elegant, I think, because it is the least experimentally complicated and relies on a bunch of interesting post-processing. And I'm going to tell you right here on this slide how PAINT works as we're applying it. So first of all, one of the nice things about ion exchange is that you can control the equilibrium and the binding characteristics by just controlling the salt concentration in the solution. And so what we do is we adjust the salt conditions and the concentration of the ligands and the concentration of the protein such that most of the time in most of the place there's nothing but agarose there, and also that the equilibrium constant is mostly on the off side, so that most of the time the proteins that are in the mobile phase are just diffusing by, and we can't see them because they're moving too quickly for us to see them, and also because we can only image right at this interface. And so due to motion blur, that's the MB part here, we're actually blind to freely diffusing proteins. But every once in a while, a protein actually binds and interacts for a longer period of time with a ligand, and then because it's within the evanescently decaying field of the microscope, it gets excited and emits photons, and then we can image that with a CCD camera, as you see here. But this is the diffraction-limited image, which means that we can't see anything really smaller than about half the wavelength of light, so our resolution here is about 260 nanometers. But this super resolution technique is really a math trick, which is one of the reasons we like it so much. One of these absorption events, you can take that image there and fit the pixel counts to a 2D Gaussian that gives you a center of your 2D Gaussian, and then it gives you a localization uncertainty in X and Y. And then you can take that information for that one event and plot both the center and the uncertainty for that one event. And then, like I said, we adjusted the equilibrium so that it doesn't spend a whole lot of time in the off state. And so when we have multiple events, we can do this statistical analysis over and over and over again. And the combination of these statistics then allows us to create a super resolved image in which we've broken the resolution of light, the diffraction limit, and gives you resolutions that approach things like 10 nanometers or even below, depending on some groups. Some groups claim to get below 10 nanometers. So that's how you use super resolution here in this particular case. And now we're able to image things that are on the order, if you're charitable anyway, of the sizes of our proteins and ligands. And so by using this method, now we can actually analyze our different samples. And this sample over on the right-hand side, and unfortunately here, because of the light and because of the resolution of the projector, you can't see this so well. On the right-hand side, this is our negative control.
Can you turn the light so let's see what happens?
Let's just see. Ah, okay, that is better. All right, thank you. Saved the day back there. The negative control on the right-hand side is our porous agarose support. What we should expect to see here, with down here on the bottom our super resolution event image, is that we should expect to see every once in a while a protein gets stuck in one of the pores of the agarose, and it's stuck for just a moment, and then it diffuses back out again. And we should expect to see, in addition that these are very fast events, we should also expect to see that these are non-repeating events. Because we have such low concentrations of our protein, we should see this completely isotropically all over the surface every once in a while, and every place around the surface completely homogeneously, that we should see one of these fast events that's based strictly on our protein getting sterically trapped for a moment in our porous stationary phase. And then in contrast over here with our clustered charge peptide, what we should expect to see is, of course, because we're still using the agarose support, we should expect to see this homogeneous, very fast steric trapping of the protein within the support. But in addition, and now this super resolution map is an event map, so it tells us the number of times that we have a particular event mapped on our sample, we should see repeat and long-lived events when a protein binds to a ligand. That's exactly what you see here with resolutions around 30 nanometers on this particular sample. But the surprising result wasn't either our negative control here or the clustered peptide. Instead, the surprising result is that under no conditions with the traditional peptide decorated within the support, and by no conditions I mean reasonable density of loading of the peptide, reasonable concentrations of the protein, or reasonable orders of magnitude tuning of the salt concentration to tune the equilibrium, never, ever, ever can we detect any meaningful repeat binding between the protein and the normal stationary phase ligand when it is completely separated and isolated. So in this particular case, our results lead us to a conclusion that's actually much stronger than the hypothesis we formed at the beginning, which was simply that clustering's happening and contributing. Our conclusion based on this data is that clustering is the only driving force that is causing the interaction between this protein and the stationary phase. So of course, that's a much stronger assertion and conclusion, and so that requires much stronger experimental evidence. And one of the pieces of evidence that's really necessary here is if I'm saying that the only way interaction with the standard ligand happens is if it's clustered, then I should be able to increase the loading density to a point where I force it to cluster and therefore force the adsorption to turn on. And additionally, if I'm saying that clusters are driving the interaction, and here I'm looking at the pentapeptide, I should also see if I move up in polymerization from a monopeptide to a di to a tri, I should see at some point a threshold where I see the adsorption turn on, if this isn't purely an electrostatic process. And so the first control then I'm going to show you is the inducing of the clustering of the monocharged peptide. We should expect to see but this is, if this really is due to clustering, and clustering is driving things, I should see two things. First of all, I shouldn't see a monotonic increase in the adsorption. I should see nothing until clustering turns on and then something. And then the other thing I should expect to see, because the ligands, the peptides are charged, I should see that it should be very difficult and it should require high concentrations to turn this on because they electrostatically repel each other, and I was working at very low loading densities to begin with. And that's exactly what you see here. If we go 10 times, 100 times, 500 times, 750 times higher loading density than in the previous experiment, we see nothing, never, never, ever, ever, any adsorption events, but only when we go about 1,000 times higher in loading density do we start to turn on adsorption with the monopeptide like we see in the pentapeptide. And additionally, we see right here that this is a very heterogeneous threshold. Some areas of the sample you see something, some other areas of the sample you see nothing, and when you go a little bit higher, that starts to even out again. And then the other thing that we should expect to see here, if I'm saying that what's going on here is that there's some sort of stochastic or random clustering that's going on that I can't see, if I measure the rate constants for these adsorption sites here, I should expect to see, if these really are due to clusters, is the distribution of rate constants should be highly, highly broadened. So I can test this clustering hypothesis further with the rate constants. So keep that in mind. The other control is consistent with our prediction, but not as convincing because of the calcium binding pocket right here. We might expect that the threshold for cluster-type behavior might be around 2 or 3, and indeed that's what we see. If I were using a bar graph here, this would be easier to see. We see nothing at mono, we see nothing at di, and then at tri it starts to turn on. And so this also is a threshold here, which is very consistent with a cluster-type interaction between this protein and the peptide. And as I said, we can treat all of these individual ligands now as individual little adsorption centers, and we can collect the rate constants for the adsorption and the desorption kinetics for all of these different sites. And for the first time, test really the first assumption of that Giddings and Eyring theory, which is somewhat of a surprise to us, but maybe things would be different if we used a more complicated protein. At every single adsorption site, we have single exponential kinetics, which is implying that there really is only a single physical mechanism driving the adsorption and desorption of the protein with each one of these sites, which means we don't have something like an induced fit where the protein is binding and then readjusting or things like that. We were expecting to see that, but we didn't actually see that with this particular protein. But then we also, with the distribution of kinetics, we can see that things are actually a little bit more complicated than what the theory predicts, and I'll show that more on the next slide so you can see it more clearly. If there were purely just a thermal distribution of the adsorption and desorption kinetics as described by that theory, all you would see here is a 2D Gaussian broadened by just the uncertainty in our ability to extract the rate constants. But instead, there's this complicated log-log inverse relationship between the adsorption and the desorption rate constants, which imply there's something more complicated going on here. And our preliminary studies on this are related very strongly to the types of things that Mary talked about before, which is our preliminary experiments suggest that this is due to the steric availability of the peptides within the agarose, which is consistent, I guess, that industries often uses these spacers to kind of even out the presentation of the ligand to the analyte. So we think that we can even get steric information about the orientation within the agarose from here. And then finally, I'll go quickly here because I believe I'm out of time. We can take that information. Okay, awesome. So one of the things we were really excited to learn is that we could then, now that we have extracted all the rate constants and time constants for these different types of supports with all the different ligands, and relate our experimental data to the theory by Giddings and Eyring, which really means, from a practical respect, that you could say that at any one of these little adsorption centers, if you created a column with that perfect interaction, just magnified it over the course of the column, you could say, via this theory and our experimental data, that each one of these is the elution profile that would result from a continuum of these types of interactions occurring through a column. And so you can take each one of these types of interactions from the rate constants and say exactly through this theory what sort of elution profile should result. And more importantly, you can compare the kinetics that you extract from our intentionally clustered or engineered clustered peptide and compare that now. Remember I said we had turned on the clustering once we went up to 1,000 times higher in loading density for our monopeptide, and we can also extract the rate constants here. Then we can say, considering all the different adsorption events and kinetics that happen on this sample and all the different adsorption events and kinetics that happen on this sample, if we combine those all via the theory, we can predict the elution profiles that will result from intentionally clustering the peptides and letting the clusters letting the peptides accidentally cluster by simply increasing the loading density of the peptides to confirm here, at least via simulations, that controlling the clustering can improve the separations efficiency here as much as 500% if you compare the relative plate heights here. But there's actually more to do. This little end peak down here is actually the distribution, the broadening that happened due to this steric broadening in terms of the availability of the ligands within the peptide. So you could do actually a lot better in understanding even these little events that lead to some of the fronting and tailing here if we wanted to, and we have plans to do that in the future. And so with that, I'll wrap up and say, given I've just started my transition, I guess, that you can use lots of interesting advanced spectroscopy to learn new mechanistic detail about processes that are important historically, but even increasingly more important to the separations community. And you can learn things about all pieces of this process. You can learn something about the mobile phase in terms of the protein and its structure, the stationary porous support, if you're interested in that, and also in the orientation and the placement of the functional ligands within that support. And those are things that we are interested in studying in the future. So with that, I will thank you and also congratulate Mary again on this well-deserved award. There is no one answer to that. The equation that describes the resolution essentially has the number of measurements on the numerator of that equation, and on the denominator of that equation is something like the brightness of the events. And so all of this is a compromise. Everything with single molecule spectroscopy is a compromise between that you want your event to live long enough for you to measure it, but not so long that photo bleaching or blinking statistics contribute to your off rates. And so there's a compromise there. If we could come up with a better fluorescent dye, then that would be the thing that would allow us to measure more events and for longer, and to get that resolution below around 30 nanometers to something more like 10 or 5. You're absolutely right. If this is purely electrostatic, you ought to be able to tune the Debye length of the solution and the absorption-desorption rate constants for these protein and peptide interactions such that you could resolve it. We did some simulations that imply that our resolution is something like 10 or 100 times, that we could even find events that were happening faster than our frame rate, on the order of 10 times faster and 100 times faster. And in addition, we integrate the signal, so we measure overall time. So even if something was happening very fast and we can't resolve it because we're summing over all time and because there's repeat, you should see a buildup of photon counts at a place where there is a ligand versus at a place where there isn't a ligand. I can simply tell you that we see no evidence of a purely electrostatic interaction between that protein and that ligand. I know that that is... Yes, we did experiments. That was one of the control experiments that we did. We changed in our manuscript, we say detectable. There's no detectable because I understand what I'm saying flies in the face of the Coulomb potential and how that should all that should imply. And we also demonstrated, so just in case we were doing something that was moving away from electrostatics, electrostatically you can tune off the interaction with the clustered peptide by increasing the salt concentration. So we are still, when we have binding, we can turn it off by using elution conditions. And so when it's binding, it is binding controllably by the electrostatics. However, it seems either it's not purely electrostatic or the electrostatics is not the primary driving force for this particular protein-peptide interaction. Yes?
It seems to me if you're load up the surface statistically with ligands, that and you start to see clusters that are close enough to, like you can develop an electrostatic model using kinetic strength sort of distance parameter to sort of map out how many are within a given sphere of influence.
Agree completely. Absolutely right. And that's also on our list of on our to-do list. And in particular, because this relates also to anti-cooperativity in binding, we would also like to kind of expand our understanding of the Hill equation to include this more mechanistic description of rate constants and how individual ligands can interact with each other. Agreed. Absolutely, yeah. And we chose to start with a simple protein and a simple ligand system, and we were surprised by that result, obviously, just as you are. I agree.